Department, Faculty of Agriculture, Asyut University, PhD from University of California, Davis in Environmental Chemistry and Agriculture. So today we have pleased actually to have Michael Mayfield. Michael Mayfield, he is actually a PhD student in Alabama University, and his research focused on Orthostosis viruses, a unique viral genus which has the ability to infect insects as well as plants and result in potential yield loss across many different groups. His main PhD research project is creating and analyzing clones of tomato spotted wild virus proteins to transfer insect cells and identifies the site of localization using fluorescent microscopy. He's also conducted the field research such as buyer surveys across growing seasons for soybean in Alabama to identify the relationship between viruses and their hosts. Uh, Michael actually his presentation today on a field and molecule approach to understanding Alabama legume viruses. So we are happy to have you, Michael. And yeah, please go ahead.
All right. Well, thank you so much for that introduction, Dr. Mohammed. So, as said, my name is Michael Mayfield. I'm at Auburn University, PhD student. And today I'd like to talk with you about my research, which I've titled A Field and Molecular Approach to Understanding Alabama Legume Viruses. So, I have two main research projects that I just kind of wanted to go over. The first being a more field-based project, which is a survey of Alabama soybean plots during the 2020 and 2021 growing season for insects and the potential viruses. And then the second project, which is more molecular based, is protein expression and localization with tomato spotted wilt virus proteins in insect cells. So to start off with a little overview of soybean, soybean is very commonly grown in Alabama. We use it for feed and also for dietary products and pharmaceutical goods. In 2019, we produced 265,000 acres, so around $135 million. And when we grow our soybean, about 80% is being used for it as a meal, meaning it's going for animal feed and some food products, while 20% of that soybean is being grown to be produced for oil, 61% for food, and then the rest going towards things like diesel and fuel. And so if you were in Alabama and you were growing soybean, you might expect to have viruses show up in your growing season. And in Alabama, the viruses you might expect to see are four big ones: soybean vein necrosis virus, bean pod mottle virus, soybean mosaic virus, and tobacco streak virus. And these viruses are vectored by insects like aphids, thrips, and bean leaf beetles. So we have a disease triangle in Alabama whenever we have a pathogen going around in the fields. And it's primarily determined by three factors like the host, the virus, and the environment. These three things can really come together and impact the severity of the disease. And so we're trying to get a better molecular understanding of the relationships between these three factors. And so to start off with the research questions, I started by asking, what viruses do we expect to see in Alabama during the 2020 and the 2021 soybean growing season? And then what insects are acting as a vector for these soybean viruses? So I chose 7 untreated plots across the state of Alabama. From north to south, this would be Tennessee Valley, Sand Mountain, Clanton, E.V. Smith, and then these two locations right here, the old rotation and Kohler's rotation, are actually both part of Auburn University. This is on Auburn's property. And then the final spot was at the bottom of Alabama called Bruton. And so the goal was to visit each of these collection sites at 2 different times over the growing season and sample for symptomatic plants as well as insects in the fields. So just to put a photo with these symptoms and these viruses right here, this first plant is soybean vein necrosis virus. You can see the necrosis happening in between the veins of the leaves. This is transmitted by thrips. We have bean pod mottle virus, which causes this distinctive bumpy and color pattern on the plant. This is spread by bean leaf beetles. Tobacco streak virus over here on the right is transmitted by thrips again. This has got symptoms that you can clearly see on the pods. It causes yield loss with the pods. And then soybean mosaic virus has another leaf bumpiness, really nasty looking leaf pattern, and this is spread by aphids. So then to match it up with their vectors, we see the bean leaf beetles, Ceratoma trifurcata, with the bean pod mottle virus, neohydrato thrips or soybean thrips with soybean vein necrosis virus, aphids in general for soybean mosaic virus, and finally we have western flower thrips, Frankliniella occidentalis, for the tobacco streak virus. So my hypothesis was viruses will appear throughout Alabama in the 2020 and 2021 growing season. These viruses are going to be those that we've seen previously: soybean mosaic virus, tobacco streak virus, soybean vein necrosis virus, and bean pod mottle virus. So if I go out into the fields and I collect tissue and perform an ELISA, I'll be able to identify the viruses present. And finally, if I collect insects while I'm at the fields, I'll be able to identify their viral vectors. So for insect collection, the way this was performed is that when I would arrive at a site, I would find 2 parallel rows of soybean, and I'd start at the front of it with the insect collection net, and I would take 100 wide sweeps while walking simultaneously down 2 rows of soybean. And then at the end of those 100 sweeps, I'd empty my net into a bag and place it on ice so that I could identify these insects when I got back to the lab. And I did 8 collections at each site. So I had 8 of these insect bags from each visit, and each site was visited twice. So 16 bags from each site containing a whole bunch of different insects. For tissue collection, when I would go out to the field, I would look for somewhere between 5 to 10 samples of tissue that I thought were symptomatic for a virus or maybe showed signs of feeding. And so right here on the left, I took these photos. I was thinking originally this looks like something perhaps like bean pod mottle virus or soybean mosaic virus, just based on the pattern of the leaf. And then on the right, I saw this, what I thought was vein necrosis, and so I assumed this might be soybean vein necrosis virus. So I was typically looking for symptoms like these when I was out in the field. And once I would have 5 to 10 bags of these, I froze them until I was ready to use them for an ELISA. And if you're not familiar with an ELISA, it's a virus detection method. I used a double antibody sandwich ELISA. The way that this works is that you'll have your well, you'll add an antibody to the bottom. After a buffer wash, you'll add your antigen or your plant sample, which sits on top of that antibody. After another buffer wash, you'll add a detection antibody. And your detection antibody, if you have a positive in that sample, you'll see a visible color change. It turns like a very bright yellow. But if there is no virus in that sample, you don't see a color change. So this allows you to get a visual identification of if a sample tested had the virus you were looking for or not. And so just so you can see what this does look like, we use a machine to make sure that we're getting good results. We don't want to, just go by your eyesight. So the machine quantifies a positive, which we see here in pink, and you can see over here on the actual plate, it's a very bright yellow. Below that is your negative. Your negative is just water. And to have a positive sample be considered positive, it must be 3 times your negative. So we see here, this is the negative, and then a positive popped up here, which was 3 times our negative. And if you look at the actual plate, you can see there's a slight change in color. So we use the plate as a way to make sure that it's not just visual bias. So how did these 2020 soybean surveys look? Well, thrips were the most collected insect, not just during the first or second, but overall. We found over 900 thrips during this first collection season. With the bean leaf beetles, we found a pretty good number of them with the 2020 survey, but we didn't find very many aphids at all during this season. We found less than 40 aphids across the entire growing season with these 2 samplings. And then how did the viruses look? So we only identified 1 sample for virus. It was soybean vein necrosis virus, and it was at the old rotation, which, just to refresh your memory, is right here in Alabama. It's actually the location of the plot that's on campus, so for Auburn University. So I thought it was kind of funny that for 2020, the only positive sample we got was literally right on our campus, none of the other extensions. So what is the conclusion? Why did we not find a whole lot of viruses sampled? Because we only found one when we ran the ELISAs, but we had undergrads doing additional tests that tested positive for tobacco streak virus. So why would they have tobacco streak virus and I didn't test it? It could come down to something like degradation of the virions. Wasn't tested fast enough, or even the plant sample that was tested, such as the leaf or the stem. But in 2019, my supervisor, Dr. Martin, observed bean pod mottle virus and soybean vein necrosis virus. So we kind of scratched our heads as to why we didn't see any real viruses in 2020. So we looked into it, and according to the National Weather Service, Alabama had its third wettest year to date in 2020. Some counties had the most rain that they've ever recorded. And these record amounts of rain allow fungal pathogens to move in. And this is what I call the year of the soybean rust. So these photos that I'm showing down below are both photos of soybean rust. And now that photo on the left is a photo of what I assumed was soybean vein necrosis virus. Because I don't know about you, but to me, I think that this vein necrosis right here looked a bit more virus-like, but it was an easy mistake to make, and we found a lot of rust. So just how wet was Alabama in 2020? So if we start by looking at the left, which is our mean temperatures, this is the state of Alabama. Our entire state was much above average or above average in terms of heat. And then when it comes to precipitation, almost the entire state was either much above average or record wettest for where I sampled. And then just to show you the typical growing season for 2020, they began in June. It went on to July, and I believe they harvested around the end of August. And we can see just a horribly wet growing season for all of Alabama. It wasn't a lot of fun. But we repeated this project in the 2021 soybean growing season. And so what did that look like? Well, we saw some similar trends, is that thrips were the most identified insect collected again. We found around 785 during the second survey and 900 around during the first survey. So thrips seem to be what we find most consistently on soybean. But we saw kind of a flip. We saw less bean leaf beetles, and we saw way more aphids, almost 300 aphids combined compared to 2020 when we only found 40. So in 2020, when we had about the same amount of thrips, we had just that soybean vein necrosis virus. For 2021, however, we not only detected every virus that we were looking for, we found virus at every single location with the exception of Clanton. And at some of these locations, we found mixed viruses. So why did we detect so much more virus than 2021, or in 2021 than compared to 2020 when we caught similar variety between insects? Well, in 2021, almost all of the growers at these extension sites were predicting another wet growing season, and they wanted to avoid those fungal pathogens and all that soybean rust. So they planted their crop in 2021 1 month later than usual. And so this change in planting time resulted in less fungal pathogens, but a lot more virus that we saw. So next steps that we are doing in the survey, what I'm currently on now. So all of those samples that I collected and used in ELISA, which were positive, I kept a bit of. I did an RNA extraction, and now I'm using this RNA extraction to get the sequence for these viruses. So my hypothesis for this is that the viruses will appear. We already know that. The viruses will be those seen in previous growing seasons. But now RNA extractions leading to sequencing of these samples will give me the virus sequence, specifically that we have in our fields. And then these Alabama sequences can be compared to identify differences or similarities to the reference genome. And so here we have a table of the RNA that I extracted from this 2021 samples. You see we have some soybean vein necrosis virus, and then some bean pod mottle virus. We also find some mixes like bean pod mottle virus and tobacco streak virus, and bean pod mottle virus, soybean mosaic virus, and soybean vein necrosis virus. So looking from here on, this next few slides might be a bit familiar if you were at Abdalal's presentation last week. So this is just a sample. I'm not actually at this step yet with the sequences, but this is a sample of what we'd like to do. So once my sequences are obtained, the next step is going to be to identify the sequence compared to the reference. And once you have the reference compared, you can see, are there consistent amino acid changes across the whole state? So this would be maybe soybean vein necrosis virus. And then one of the samples we find, are there consistent amino acid mutations that are happening that makes the Alabama strain specific? Maybe these mutations are even causing something like resistance breaking or helping the virus infect the soybean. And also, once you have your sequences, you can do some fun stuff like throw them into phylogenetic trees and see where do your sequences line up with the soybean that's grown in neighboring states like Georgia and North Carolina. You can even add in some diversity with Asia. So future studies, this experiment will be repeated over several consecutive growing seasons to continue to monitor which viruses and insects appear over time. And it can also determine how changing management conditions influence these insect populations, such as planting earlier and later in the year and what viruses you might obtain. And then finally, you can identify key changes in the amino acids from these Alabama strains compared to reference. So that was my first research project, the field-related one. We're going to get a little bit more molecular with this next one. We're going to be talking about protein expression and localization with tomato spotted wilt virus proteins in insect cells. So we just talked about soybeans. Let's talk about another big legume in Alabama. We grow a lot of peanuts. We harvest about 400 million pounds of peanuts every year, bringing in about 200 million for our economy. Now, you might be wondering, who are we selling all these peanuts to? Because we're shipping them out across the world, but one of our largest buyers is actually Mars Chocolate, the people that make delicious things like Snickers and peanut M&Ms. They're buying these really nice high-quality peanuts to put in their candy bars to make them as good as they can, and it's projected that they're going to want to triple their need for these high-quality peanuts by 2025. So tomato spotted wilt virus is a virus that's got this huge, incredible host range. It can infect over 900 species of plants. But in Alabama, peanuts are a primary concern. And this has been a virus that in the past has not only been found in Alabama, but it's caused a lot of damage. From 1986 to 89, 288 peanut fields were surveyed, and 82% had plants that tested positive for TSWV. So this is a virus that can be a threat to our peanuts, and we don't want anything to do with it. Now, we have resistant strains against TSWV, but with viruses always having the ability to mutate, it's always the best to know as much about the virus as you can before something catastrophic happens and we're playing catch up with our knowledge. TSWV, its genome, is divided into 3 segments. They're very nice how they named these. They named them L for the largest segment, M for the medium-sized segment, and S for the smallest segment. On your L, it's going to encode a polymerase protein, while the M segment has 2 glycoproteins and a non-structural movement protein. The S segment has a nucleocapsid protein, as well as a non-structural silencing protein. And so TSWV is in the genus Orthotospovirus, which is order Bunyavirales. And this is really important and unique because these viruses can cause infection in both animal and plant cells, which is a trait only shared by one other order of viruses. And the ability to cross the kingdom barrier means that there's some differences in cell types that these viruses have been able to overcome. So just a quick refresher, animal cells and plant cells, one of the biggest differences is that our plant cell has a cell wall. Animal cell does not. Plant cells also have the plasmodesmata, which they use for that intracellular transport communication. Animal cells do not have plasmodesmata. And then the final big difference is that plant cells typically have this large central vacuole, and there's not a large central vacuole in animal cells typically. So I told you it infects animal and plant cells. The animals that it would infect are thrips. It's transmitted by at least 11 species of thrips, including Frankliniella fusca, Frankliniella occidentalis, and Frankliniella schultzii, which are 3 big ones for Alabama. The thrips transmit this in a very interesting way. I'd be not telling you something very cool if I left this out. So to transmit tomato spotted wilt virus, the thrips have to take it up into their larval stage. So these are their first 2 larval stages. These are their 2 pupal stages. And finally, there's a male, and then there's a female. So it starts when the larval stages eat an infected plant. They take the virions into their gut, and then when they enter their pupal stage and they're metamorphosizing, they become, for lack of a better word, gooey on the inside, and their salivary glands actually press up against their gut. And what happens then is that all of the plants that they eat as an adult, they'll be salivating the virions into the healthy plant. Now, if they were to eat an infected plant as an adult, they can take the virions into their gut, but they're not able to transmit the virus. They can only transmit the virus if they acquire it as larvae, pupate, and then afterwards they'll have the virions in their salivary glands. So I think that's a really cool mechanism that the virus has evolved millions of years more than likely to be able to have with these insects. And so TSWV proteins in the replication cycle for the plant cell. When I started this research, a good bit was known about the plant cell, but really not much was known about this virus in insect cells. So just to go over those proteins again, the nucleocapsid protein, its goal or its function is to encase genomic RNA. The non-structural silencing is going to be a suppressor for RNA-mediated gene silencing. This non-structural movement protein in plant cells forms tubules through the plasmodesmata, so that's really interesting. And then both of the glycoproteins are used to help with receptor-mediated endocytosis. So how does replication cycle take place? So it attaches in a cell, entry through receptor-mediated endocytosis, uncoding will take place before primary transcription. You'll get translation of the L, M, and S mRNAs before you get membrane-associated RNA replication. Lead down into the Golgi for morphogenesis and glycolysation for the glycoproteins. And then finally, you migrate out of the Golgi vesicles to the cell surface where they get released as infectious virions. Now, plant cell microscopy has been done for tomato spotted wilt virus. This was my supervisor, Dr. Martin's work. So in plant cells, we see that the nucleocapsid protein creates aggregates in the cytoplasm. Let me explain these first. GFP is going to be tagged to the protein so that the left column you can see protein expression. Middle column is RFP, which is used as a a free cell marker to allow you to see the cell better. And then an overlay shows you where is that protein expressed when compared to the exterior of the cell or the cell itself. So when we look at the nucleocapsid protein, we find it forming aggregates in the cytoplasm. The non-structural silencing protein, we find it in the cell periphery. We can't actually say cell wall because you need specific tests and markers for the cell wall. In SM, we find actually right at the plasmodesmata, since it's forming tubules to go through the plasmodesmata, we find it linking cells. And then both of the glycoproteins are found in the cytoplasm for plant cells. So research questions that I had for this is, how does the localization of these viral proteins compare to what is known about the virus? And how will identifying the location of the expression in insect cells compare to the location for the proteins in plant cells? So my research goal was to take those tomato spotted wilt virus open reading frames, all of the proteins except for the polymerase, and then fuse them to sequences for either GFP or RFP. Then I wanted to take those constructs and use them to transfect SF9 insect cells. We're using SF9 insect cells because there is no thrips cell culture. SF9 cells, however, have well-developed protocols and they work great with the promoters and vectors I used. And then once I've transfected these cells, the goal is to perform fluorescent microscopy to perform and identify where those viral proteins are localized in SF9 cells. My hypothesis is that these proteins are going to be in different parts of the insect cells relating to the infection cycle. We know where these proteins are localized in plant cells, so it's hypothesized they'll be in similar locations in insect cells. These patterns will likely correlate to the locations such as silencing, replication, and movement. And finally, localization of each protein alone may differ from localization of proteins expressed together. This is a goal that's way down the line, though. So how did I create my constructs? I started out with a p-enter d-topo entry clone, which had my TSWV proteins, and then I recombined it with a PIB destination vector, which contained either GFP or RFP in 2 orientations when possible. I'll explain that in a second. And so what I was left with was an expression clone, which had my TSWV protein tagged to a fluorescent marker, and we got rid of a toxic byproduct. So I mentioned I tried 2 constructs when possible. What I meant by this is that I had what were known as PIWG vectors, where the gateway cassette was before the floor, as well as PIGW vectors, where the floor was before the gateway cassette. And we tried it in both of these orientations to see, did we get a difference in expression or did it prevent expression? And then if we did get a difference, we wanted to take photos with the one that was better, of course. We also tested using GFP and MRFP to see if either one of these would be better with fluorescent. And so when I had the gateway cassette before the floor, there was NSS, NSM, GN, as well as a soluble form of GN, GC, as well as a soluble form of GC. And the reason that we created soluble forms of these is that we know from the plant microscopy experience, it can be very difficult to get either of the glycoproteins to localize just by themselves. So we tried a soluble form in hopes of that giving us some fluorescence. And then when it came to the floor before the gateway cassette, it was just the N, NSS, and NSM protein. We didn't have to worry about the glycoproteins in this orientation, because they have a signal peptide and transmembrane domain that would prevent them from ever being like this in nature. So as I was making these constructs, I was transforming with top 10 cells and doing colony PCR to confirm my gels. And not only was I just going off my gels, I was sending results to Eton Bioscience for sequencing, and they were sending me back that everything was good. So doing cell passaging was something that was required for transfection. You have to grow your cells in order to transfect them. The SF9 cells grow in the bottom of the flask, and about once or twice a week, they have to be passaged to a new flask to prevent cell death at overpopulation. And sanitization is key. It took me a while to get good at this, but this is typically what it looked like when I was passaging or getting ready to passage to begin a transfection. And so what's the transfections? It's a way of essentially getting the plasmid into the insect cells. The way we do this is we take a reagent, in this case lipofectamine, you combine it with your plasmid, it forms a complex. This complex is added to the cells and it actually can enter the cell and form an endosome. Once the endosome is in the cell, it starts to destabilize and your plasmid is released. Your plasmid localizes to your nucleus and the gene expression cassette is transcribed. So from then on, all cells replicating from those cells will have your with the protein and the fluorescent promoter active. So I had to make a bit of constructs, as you see here, for GFP N1, where the floor was before. We had all of these constructs as well for RFP, while we didn't have to worry about the glycoproteins with the rest. So this ended up being a bit of work. There's 20 constructs total. I needed 3 successful transformations for 3 different replicates, and then each replicate needed 3 publications here photo sets. So it ended up being around 180 photo sets. So let's get to the results. The first thing that we see is that there was no difference between the gateway cassette being before or after the floor. So we see here the protein on the bottom is before the floor, and the floor is before the protein on the top. Localization is identical. It doesn't change at all. We also saw that there's no difference between GFP or RFP. The only difference was an aesthetic color difference. Localization was identical every time. So insect microscopy results, I'm going to sum it up kind of briefly, and then we'll go over them in detail in a minute. But to sum it up briefly, these were all found in the cytoplasm. So GFP is going to show us where the protein is localized. DAPI is our nuclear marker, and then an overlay so we can see. So for the N protein, it's cytoplasm, and we see it on the outside of the nucleus. NSM we also identify in the cytoplasm, but it has another kind of similar to N outside of the nucleus pattern. NSS cytoplasm, and we see it in the cell. All of the glycoproteins are found in the cytoplasm as well. So you can see here. And so how does this compare to the plant microscopy? So in insects, we find the nucleocapsid in the cytoplasm, and in plants, we also find the nucleocapsid protein in the cytoplasm. This makes sense to me because you would want the nucleocapsid protein to be one of the first things to interact with anything leaving the nucleus as it encapsulates that genomic RNA. In NSM, we find it in the cytoplasm, while in plant cells, we find it in the plasmodesmata. Well, insect cells don't have plasmodesmata, so it makes sense to me that this protein might be specifically plant-based, and that's why we would just find it in the cytoplasm. We see something similar with non-structural silencing protein. So NSS, cytoplasm for insect cell, but in the plant cell, it's actually pushed along the cell periphery. So that large central vacuole in the plant cell is pushing it to the periphery. But again, insect cells don't have that large central vacuole. So it makes sense to me that you would also find this in the cytoplasm. And then for both plant and insect cells, GN and GC were found in the cytoplasm. We were only able to get GC when it was soluble for both plants and insects. That's how difficult it was for that protein. And further research would be done with the glycoproteins for Golgi markers because we know that they do associate with the Golgi during infection. And then after looking at these two insect protein localizations right outside the nucleus, I think this is almost suspiciously something that could be Golgi. So I'd like to also in the future do Golgi markers with these proteins as well. So as we see, all of these proteins were found in the cytoplasm with the exception of NSM being a plasmodesmata in plants and NSS, this non-silent structuring, getting pushed to the periphery in plants as well. So conclusions and next steps. Now that individual protein localization has been performed, proteins can be transfected together to see if localization changes in the presence of other proteins. And so doing this would put together a protein interaction map, which can identify what parts of the cell the proteins occupy in the presence of each other, because maybe N is in the cytoplasm by itself, but when it's expressed with NSM, we see it doing something like go to the nucleus. So future research could be done with interaction mapping, and also we could try doing individual protein localizations with another orthotospovirus, such as soybean vein necrosis virus. And this is something like what that protein interaction map would look like. We would be testing all of them against each other in the future. So significance of results for this section, the first question to be asked was, what is in our fields? So we went and identified it. The second question asked, what is the sequence for the virus or viruses found in our fields? So that's part of my current work. The third question is, where are the proteins expressed in insect and plant cells for a particular virus of interest? And then if you know, not just TSWV, but if you know where these proteins are expressed by themselves and co-localized with other proteins, you could use this knowledge to try to disrupt or stop the virus from infecting the plant or insect cells and prevent infection. So that being said, I'd like to thank everybody at MGBS for having me. Thank you all for listening. I'd like to thank my supervisor, Dr. Martin, funding, which is from the Soybean Farmers Association and the Department of Entomology and Plant Pathology. Thank you all, and if there's any questions, I could take them now.
Okay, thank you very much, Mike. Really valuable presentation and a lot of information too. Actually, if it consider it in Egypt like a key pest, transmitted many disease, like a bunchy top virus in banana and mosaic virus, especially in vegetable crops, which make aphid like a double risk in Egypt actually. So thank you very much. And now we open the question. Anyone have any question? So don't be shy. So yeah, there is a question actually in the chat room from Shirihan El Amin. She asked you if that's possible, of course. Do you have like a papers published from the work or papers? So she asking if that's available. Yeah, she need the papers. Yeah.
We just finished the final draft of it on Friday, and
Okay.
we're sending it to the lab where it will be coming out of. It's going to be a big paper that shows pretty much it's the first orthotospovirus full look at the plant protein localization and the insect localization. And it goes a bit farther. They do a yeast 2 hybrid to actually identify the interactors in plants. So I don't have the publication right now. If you'd like to give me your email, I could take it down. And once it's published, I'd love to send it to you.
Yeah, that will be great. Yeah. So, any other question?
If there are any other questions that you guys think of later, I have my email. I don't know if you can see it here at the bottom, mam0279@auburn.edu. So you can send me anything. I'd love to talk about any ideas.
So yeah, if you have any questions, you can directly contact Mike and ask him anything. So yeah, there is another question from Department of Genetics in Asset University. Are there specific genes in soybeans that are known to confer resistance or susceptibility to the identified legume viruses? Actually, the question in the chat room too.
Okay, yeah, that's a very good question. I don't know if there are any for those 4 viruses that I've spoke about, but that's a good point. It'd be good to look into that and see if there are any so that we could potentially try looking for interactors with that.
Okay. Any other question here? Okay. Okay. Yeah. Yeah. So Abdulal Hammam, he wanna ask a question here. Okay, Abdulal.
Hello,
Yeah. Go ahead.
you hear me, Alec?
I can
Yeah.
hear you.
Okay, so thank you so much for the very valuable presentations, and I just have one question. So does the localization in the insect cells can tell us if the protein is interacting with something from the insect or not? And if not, how can we know?
So that's a good question. I would say based off of the microscopy that I showed here, where we're just trying to do a general localization, we can tell if it's in the cytoplasm or the nucleus from this type of study. The next study, which I'd like to do, would probably need a confocal microscope so that we could see organelles better, as well as organelle specific markers. So I would like to redo this using a Golgi marker, an endoplasmic reticulum marker, and I think that would be the best way to see if they're interacting with specific organelles on their own. Did that answer your question?
Yes, part of it. And then the other part is like, yeah, that would tell us, like the confocal would tell us if the protein is interacting with the organelles. But how could we know if the protein of the virus is interacting with another protein in the Golgi or in the nucleus or somewhere, like protein interaction between the viral and the insect cells? How could we know that?
I think the best way that you could do something like that would be to do like a yeast 2 hybrid to try to identify the insect interactors in the insect cells. So just doing like a yeast 2 hybrid with a bunch of proteins in the insect cell or things that you think the virus would be interacting with for each of the proteins to see if any of the markers that you pick were specific.
Yes. Yeah. Thank you so much. That answers
You're
my
welcome.
question.
Thank you.
Thank you, Dr. Michael. for this informative lecture, and see you
Well, thank you.
in other lecture for MGBS.
Great. Thank you so much for having me.
Yeah, I don't have any questions here in your room chat. So thanks a lot.
Thank you.
Okay, Michael. Yeah, sorry, we have like a problem with the connecting. So yes, thank you very much, and we are looking forward to seeing you again in MGPS seminars. And thank you, everyone. And please tune with us for the next Wednesday for another valuable presentation. Thank you, everyone. Thank you, Mike. And
Thank you all very much.
Have a great day. Okay. Bye-bye.
Thanks. Have a nice day too.
Okay, bye-bye.